Biology 104, where we will discuss plant diversity in the seed plants. The gymnosperms are currently represented by four main phyla. The first phylum, Cicadophyta, are plants that have extremely large cones, depicted in the image below, and palm-like leaves. They also have flagellated sperm, and most species are considered to be threatened or endangered. The phylum Ginkgophyta have deciduous leaves that turn gold in autumn and can fall off. They also have flagellated sperm. This is currently represented by only one species, Ginkgo biloba. The phylum Cnidophyta have very diverse shapes in their plant structure. They form pollen tubes for fertilization and thus have non-flagellated sperm. They're currently represented by three different genera: Cnidum, Ephedra, as well as Welwitschia. And finally, the last phylum is phylum Coniferophyta. This is the most diverse phylum of the gymnosperms. Most of these plants have woody cones, however some will have fleshy cones. Most are evergreens and few are deciduous, losing their leaves in certain seasons. These include your conifers, pines, firs, as well as redwoods. We're now going to discuss the next clade in the seeded plants, the angiosperms. Remember that these plants have two key adaptations in addition to all the derived traits discussed previously. Angiosperms have flowers and fruits as their reproductive features. Angiosperms are now considered to be the most diverse and widespread group of plants that dominate today's landscapes. They now include over 90% of all plant species. However, all of these plant species belong to one phylum, Anthophyta. Flowers are considered to be highly specialized structures for reproduction. They can be pollinated by many different things such as insects, animals, as well as wind. Flowers typically contain up to 4 different types of modified leaves called floral organs. These include the sepals, which enclose the flower, the petals, which can or cannot be brightly colored, usually dependent upon the pollination mode, the stamens, which represent the male reproductive organs, and the carpels, which represent the female reproductive organs. The stamens are the microsporophylls that end up producing the pollen grains that contain the male gametophytes. The stamens consist of a filament, which is a stalk-like structure, and the anther, which is the terminal sac that produces the pollen granules. The carpels, which represent the female gametophytes, are also called the pistils. They contain the megasporophylls that produce the ovules that eventually contain female gametophytes. The carpels consist of the stigma that collects the pollen, the style, which is the stalk, and the ovary, which can contain one or more ovules. This slide here depicts the overall structure of the flower. Note that in the center of the flower we have the carpel. The carpel contains the ovary, style, as well as the stigma. Note within the ovary we have the presence of multiple ovules. These represent the female part of the plant. The stamen represents the male part and consists of the anther as well as the filament. These are enclosed by the petals and also the sepals. Within the flower you can have different types of symmetry: bilateral symmetry as represented in the orchid, or radial symmetry that is represented in the daffodil. Another characteristic of angiosperms is the ability to produce fruit. After fertilization, the ovary wall will thicken around the seed to protect it. This will allow for the ovary to mature, which eventually forms fruit. Fruit can be fleshy, as in tomatoes, or dry, as in nuts. Fruits act to protect the seeds and also to provide a mode for dispersal. As mammals and other herbivores eat this fruit, they will then pass the seeds within their digestive tract, which usually will then get passed out in the feces. Plants can also have different types of adaptations to help disperse their seeds. These can help the seeds be carried by animals, wind, or water to new locations. Within angiosperms, The male gametophytes are located in the pollen granules, as we see in the gymnosperms. However, in this instance, they're produced by the sporangia of the anthers. The female gametophytes will develop within the ovule, which is called the embryo sac. This is present at the base of the stigma. Many flowers have mechanisms that help to maximize cross-pollination between different plants of the same species to maximize their genetic diversity. If a pollen gland lands on a stigma and germinates, the male gametophyte will extend a pollen tube down towards the ovary. This will eventually penetrate the ovule through a pore that is termed the micropyle, similar to what we saw in the gymnosperm life cycle. Double fertilization can occur if the pollen tube releases 2 sperm within the ovule. Once fertilization occurs, one of the sperm will fertilize the egg while the other combines with the 2 nuclei in the female gametophyte. This initiates the formation of what is termed the endosperm. The endosperm is important as it will nourish the developing embryo. The embryo itself consists of 2 main parts, the root and the cotyledons. The root is a primary root that typically develops into a taproot. This is helpful in anchoring the seed during germination after it disperses. The cotyledons contain 1 or 2 seed leaves to ensure early photosynthesis so that the developing plant can carry out photosynthesis to make important organic compounds. This diagram represents the typical life cycle of an angiosperm. We begin this cycle by examining a mature flower present on a sporophyte plant. Note that this is considered to be diploid. Within the flower, you will have the male and female reproductive organs, represented by the carpel, which represents the female reproductive organ, and the stamen, which represents the male. Within the female reproductive organs, you will have the presence of the ovary at the base of the carpel. Within the ovary, you will have multiple ovules. The ovules will house the megasporangium. Within the megasporangium, you will have a megasporocyte that will undergo meiosis to form megaspores. These will then be surrounded by the integument that will also have the micropyle, which will serve as the site for the pollen tube attachment. These megaspores will develop into a female gametophyte that will be present within the embryo sac. If we go back up to the top of the diagram, we will now take a look at the development of the male pollen grains. Within the stamen, you will have the microsporangia present in the pollen sacs. Within the microsporangia, you will have microsporocytes that will undergo meiosis to produce the microspores. These will eventually form into pollen grains. These pollen grains will house the male gametophyte. When these pollen grains disperse, they will attach at the top of the carpel, at the stigma. They will then produce a pollen tube, which will then deliver the sperm into the ovary of the plant. As the pollen tube extends, two of the sperm will travel down. Once they meet an ovule, they will attach at the micropyle site. The discharged sperm nuclei will then enter into the egg. One of these sperm nuclei will fuse to the egg to form a zygote. The other one will house and develop into the endosperm. The endosperm will serve to nourish the developing embryo. Also note the presence of a seed coat that will help to protect the developing embryo. This will then develop into a seed. This seed will then fall into the surrounding soil and germinate to produce a mature plant. The exact evolutionary history of flowering plants is still yet to be understood. We know that the first angiosperms begin to appear in the fossil record approximately 140 million years ago. The earliest pollen fossils are found within the fossil record approximately 130 million years ago, and the earliest evidence of flowering plants appear approximately 125 million years ago. Molecular and morphological evidence suggests that living-day gymnosperms diverged from a lineage that eventually led to angiosperms, and this occurred approximately 305 million years ago. Benedictalis is considered to be an extinct group of plants that had flower-like structures. However, these flowers lack carpels, true flowers, and did not have any vessels. Researchers hypothesize that these early angiosperms were woody shrubs with small flowers and had simple water-conducting cells, thus were the beginning of vascular as well as flowering plants. This image represents the current phylogenetic hypotheses regarding the evolution of angiosperms. Note two important events: the common ancestor and divergence point that occurred approximately 310 million years ago, that represents the divergence point between extant gymnosperms and angiosperms. Also note the presence of the most recent common ancestor approximately 140 million years ago. This is considered to be the most recent ancestor for all living angiosperms. The living angiosperms are represented by Amberella, water lilies, star anise, and relatives, as well as magnolids, monocots, and eudicots. Also note the extinct representative Benedictalis. This picture on the left represents the flower-like structure present within that species. Note the presence of the microsporangia, the ovules, as well as the flower-like shape. Within the evolution of angiosperms, we typically see a very strong evolutionary relationship with various types of pollinators. Many times, the pollinators can influence the evolution of the plant and vice versa. For instance, predation by herbivores can exert a very strong selective pressure for plant defenses. In addition, there is typically strong selection between pollinators and flowering plants for mutually beneficial adaptations. This close association between plants and their pollinators can also lead to speciation. An example of this relationship involves the honeybee. In order to harvest the nectar, the honeybee must land in the flower as shown. This position causes the flower stamens to arch over the bee and cover the abdomen of the bee with pollen. This pollen will then be transferred to plants within the same species, increasing the genetic diversity and reproductive strategies of the plant. Angiosperms are currently represented by more than 290,000 living species. Angiosperms are commonly classified based on their cotyledon structure. The previous classification system classified angiosperms as monocots containing one cotyledon or dicots containing two. However, recent DNA evidence suggested that the species that were classified as dicots were actually paraphyletic. This has led to a recategorization of the dicots. The dicots now form a large clade that is known as eudicots or true dicots. The remaining species that were classified as dicots now form four small lineages: Amberella, water lilies, star anise, and their relatives are considered to be the basal angiosperms that diverged early in the phylogenetic tree, while mangolids diverged later. This chart here represents the typical characteristics of monocots and eudicots. Monocots will have one cotyledon in their embryo. If you examine their leaf venation, the veins usually run parallel. Their stems have vascular tissue that are in an unpatterned scattered type of arrangement. Their root system usually contains fibrous roots and do not contain a main taproot. Their pollen consists of a pollen grain with one opening, and their flowers are usually arranged in multiples of 3. Eudicots, on the other hand, have 2 cotyledons in the developing embryo. Their leaf venation is usually in a net-like arrangement, and they have highly arranged vascular tissue. Eudicots typically have a taproot that serves as the main root, and their pollen grains usually have 3 different openings. Their floral organs are usually arranged in multiples of 4 or 5. Angiosperms are currently represented by 4 main groupings. The basal angiosperms include the first lineage, which is represented by Ambarella trichopoda. Two other lineages are believed to diverge later, and they're now represented by two clades, the water lily, as well as the clade that contains star anise and all the relatives, as depicted in the images below. The second main grouping is the mangalids. These include magnolias, laurels, and black pepper plants. It is believed that the mangalids are more closely related to monocots and eudicots than they are to the basal angiosperms. The third main grouping are the monocots. These include species such as the orchids, grasses, and palms. And the last grouping is the eudicots or true dicots. These include the legumes, roses, ornamentals, fruits, as well as flowering trees. Angiosperms have many uses. Seed plants are important for food, fuel, wood products, as well as medicine. Most of the calories that we consume are derived from 6 main crops: maize, rice, wheat, potatoes, cassava, and sweet potatoes. The agriculture industry is dominated by domesticated plants that we have produced as a result of artificial selection for desired traits. Flowering plants can also be used as tea, coffee, spices, and cocoa. Habitat destruction is the leading cause for many of our plant extinctions. Much of our rainforest is being cleared each year. At this rate, we would lose all of our rainforests in 175 years, which is not a lot of time considering the prehistoric record. Forest loss reduces the absorption of carbon dioxide from the environment and puts many other species at risk. It also can leave many medicinal compounds undiscovered. Note the pictures below that indicate the damage as a result from clear cutting of the tropical forests in Brazil. Thus, it is very important to protect our plant species to maintain the integrity of our ecosystem.